pleasure to see so many faces. And as I said yesterday, it's a reincarnation of Coffee Buddies, which was such a successful period of time during or events that we put on during the COVID experience. And you know, our goal of Coffee Buddies is to entertain, engage, and inform, which we always say. But I think it's also this thing about increasing the probability of serendipity working for you, which is, I think, a very interesting. You know, they never told me that on the MBA courses I used to teach on, that serendipity was probably the best form of business development, but it probably is, especially in today's world. And it's always a pleasure to introduce interesting people to talk to. And today we have Joanna, which is a fantastic chance for me to find out about Joanna. And I'm going to dive straight into it and say, Joanna, who are you? What do you do? And tell us something about the business that you're involved in.
Thank you. I'm Joanna Barbara. I'm a bit of a jack of all trades because I was a scientist originally and grew into leadership and business. So now I moonlight, right? Like many of you. I work for BioIVT now, but I came from legacy Xenotech, and we specialize in vitro drug-drug interaction work. BioIVT provides products and biospecimens across the gamut of life sciences research. And funny enough, I'd worked with them in a previous life when they were bioreclamation. Of course, I was bioanalysis, so I always bought my stuff from them. But about a year ago, they bought us from Xenotech, where I'd been working for 15 years. And so that's why Darina and Amola and I all worked together, and how they introduced me to Coffee Buddies. I've been with Xenotech Legacy for about it's actually 16 years. I keep telling everyone 15, but it's 16 this month, so long time. And I started out in mass spec and grew the MedID business for Xenotech, and then grew into the various other aspects of the business that we run now. Fantastic.
I mean, 16 years. How would you describe the current biotech space that you operate in, and where would you say it's been changing in the last 5 years?
So that's a great question. The biotech space I operate in is a pretty exciting place to be right now. I'd say we're a lot more. So Xenotech was always very data driven and solutions driven, but I think the industry as a whole is kind of edging more towards data driven and sort of more useful data, if you will, making a lot of the data and really considering what we're doing. I think things are a little tougher right now economically in some of the space, and so that kind of causes you to really have to think about what you're doing. And I'm seeing that a lot with the clients. They're looking for solutions. They don't want to just throw everything at the wall and hope something sticks. It's an interesting place to be.
Do you tend to work in the discovery phase of drugs?
So we are between discovery and development, I would say. Technically, we're in the development space, but we're pre-IND mostly. And so just before you get into human is where we typically encounter our clients. So they finish discovery. If you've got a ton of molecules, you're not going to work with us. But if you've got a lead compound that you're trying to package for IND right now, that's what you're trying to do. You're trying to sell to somebody else or something, and you want to understand in vitro DDI, drug-drug interaction liability, that's where we come in. So we're sort of just on the cusp of the beginning of the longer term tox studies before you submit the IND package. That's where the majority, maybe 70–80% of our clients are. But we also on the back end with post-marketing commitments. So we kind of float back and forth. And if something weird happens in clinical, you know, and they're worried about a metabolism liability, they might come to us for some in vitro support.
And what about, I mean, I'm very interested in, I've always been interested in the Gartner curve or the Gatner curve, this fast adoption and then how you scale up. And then there's this book somebody's brought to my attention called Crossing the Chasm, which again is about how sectors take on new technology. I mean, how are the customers, how are you seeing them take on new ideas? Because you're promoting some new ideas, I presume.
Yeah.
So how do they take on those ideas?
And it all depends on the mindset of the client. They come in different kind of categories, if you will, not to categorize people, because of course you can't.
No, but it's good to do that. It's good.
But you kind of have to in order to make some, you know, when I come into a client's conversation and they're just throwing all this stuff at me, right? You walk in and they tell you everything they know about their compound if they really want help. If they don't really want help, they're going to say, I want you to do this study, I want you to do it this way, and I'm not that interested in what you have to say about it. But the ones that I really love helping are the ones that I've got this compound, this is my problem, this is what I'm worried about, here's my exit strategy. And so the interesting bit about it is the educational piece. You get to really have a talk about what things mean. So some of the work we do is sort of pretty, it's almost, you almost don't have a lot of new tech going on in it because it is designed to fulfill regulatory needs, right? So you're going to do it a certain way. So where it gets interesting is the compounds themselves and sometimes the disease states themselves. I was really interested in the medical device for Alzheimer's mentioned earlier, because that's a tough disease space to be in.
I think this is really interesting. This, because I came to this world a lot from clinical CROs way back in the 80s, but I mean, what it's become more is more consultative experiences, and it's very interesting how you create that consultative thing, isn't it? Because the buyer has to sort of almost say, you know, help me, which is different when you're then picking a menu of activity.
I think the funniest part of it is when they're not quite saying help me because they're not quite ready to say help me, and you kind of have to guide them to help me, because I do feel like there's pressure on people now to be able to do a lot of things with less in terms of resources. And so because of that, they almost feel like they kind of have to have the answer, they got to move forward, and you just one of the reasons I was excited to do this was because I think talking to people about what you do is how you build some of those bridges, how you make some of those connections of, oh my goodness, I don't know this. But in the CRO space, what certainly people at medium or big pharma may not see, I don't have a deep knowledge of the cardiovascular space, but I can help you with your compound problem in the drug-drug interaction world because I've seen hundreds and thousands of compounds before. Odds are I've got some ideas. That's the fun part.
And are you seeing molecules that are more difficult now than they were? And how would you describe the difficultiness?
So a lot of the easy stuff has already been done, right? We've already done it. And half the molecules just made it to market before we even did some of this testing way back. So now there's a lot more disparity in molecular chemistry. I actually spent quite a long time last night reading about monoclonal antibodies, molecular chemist, right? Monoclonal antibodies and drug-drug interactions, because I get a lot of requests about that, but the fact is they don't interact with enzymes and test systems the same way as a small molecule. So we spend a lot of time thinking about these things. And okay, so what are you going to do then? And of course, I'm bioanalysis and mass spec really, and I have a little bit of protein experience, but predominantly small molecules. So it's kind of a different world. The really difficult thing is it worth doing the same test anyway, because these don't operate the same way? And if you are going to, how are you going to modify and how are you going to measure those things? And what are you going to make of the data at the end? Big challenge.
I mean, I often worry that we're producing too many things that will cause marginal benefits to patients.
That's probably fair.
And I mean, I suppose we're back to not the easy things, but the pivotal biological systems, by and large, we've been pivoting around them. In other words, we've hit the, if you like, the big nodes of activity. So now we're moving to the smaller nodes and hoping that they work upstream. So I suppose the question is are you seeing lots of small molecule work, or are you, what's the fashions that you're seeing in the customer base, really? What is the fashions you're seeing? Is there an area that...
No, I love that question, actually, because that's exactly how I think about it. So a lot of small molecules not dead, it's not going anywhere. We're just after different diseases and things, which is great because we know how to handle those. But investors particularly, I'm obviously in business land a lot of my life now, and marketing and sales reps and all of that. When you're out on the road, you see a lot about large molecules and biomolecules, right? They're all over the place because a lot of the recent growth and what we're talking about is large molecules. But it's about the same percentage it's been in terms of what actually comes through for many years, because a lot of molecules make it through IND because most of them fail at efficacy, right? So for me, I see a lot of the same, but the fashionable, the cool stuff that people want to talk about is predominantly large biomolecules, I would say at the moment, which is really tough in my space because, you know, they're not going to interact with the CYP.
But it's very interesting that you say that it goes through to efficacy. It's because it's very interesting, isn't it? Because with a large molecule, it's interesting about therapeutic windows and risk-benefit rate. Because in a way, what we're saying is that, you know, that because they're highly targeted or they're hitting the immune system in some way, that you don't have to, I wouldn't say you don't have to worry about the toxicity, but you're more likely to get it through. And then you've got, but so the hit is in the risk, which is interesting because you might end up with more late stage failure, actually.
I actually think that's true. And one of the big challenges, right, with large biomolecules is there is this whole immune response cytotox, giant potential. But the real problem with it is that you can't test it except in vivo, really. If you really want to know, you've got to get into non-human primate or human. And human is obviously very, very risky. And the whole non-human primate industry is really challenging right now because of the shortage of research animals. So it's a tough one to get all the way to market. And you know, when you're putting that kind of burden on the patient and the doctor of infusions and things, that's an expensive undertaking. But you really want to know it works. You do have a bit of a leg up though in terms of, you know, it's going to bind to your target, right? So that is the upside of this.
Yeah, I mean, if you were to put a bit of a crystal ball on the table and look to the next 5 years through your own lens of your own business and what you know, what do you think? How do you think it's going to mature? Is there any insights that you have into what the next 10 years might look like?
I think things are going to get more competitive, I would say. I think there's going to be a lot more push to be even faster. And I mean, that's a bit cliche because we're always pushed to be faster, but I think that it's really going to come down to that because I think resources are tightening a little bit, at least for 3 years or so. As far as actual molecules, I don't think small molecule goes away. I think there's still a contingent of small molecule that you're developing, perhaps a little bit more of large biomolecule. What I think is interesting right now is I'm seeing more sort of siRNA type things. What I would think of, that there's a bunch of sort of mid molecules that aren't quite large molecules because little guys, but they aren't really small molecules. How do you treat them? And I think that's the space we're going to move into because the challenges of both sides are a little bit mitigated. So I would expect more of that. And I am seeing growth. I'm seeing more inquiries. The challenge is people don't know what to do with them. You know, do you still have to?
It is interesting because in a classic market, you would have more market attrition. In other words, the market would say, we're not interested in this. But because of the length of the life cycle of drugs being 7 years or more sometimes, market forces aren't able to operate in the same way. You know, so you don't get, if you like, the early deaths because the market doesn't want it. It just carries on, and then you end up with more and more molecules coming on around. But the market forces don't really operate in the same way as if you're having a car or something.
No, they don't. And it's interesting because I think it's different in Europe versus the US too, because you've got all these influences on the market in the US from the whole insurance industry that's a little bit different too. Yes. So if there's a drug that's going to be a cheaper burden on the insurers and things, you know, it's going to get quite a lot of help in this country.
Yeah.
So the market forces are very specialized for the industry that we're in.
Yeah, no, that is really interesting. Anything else you would like to tell us about your own journey or the journey of your company or its product. I'm really enjoying talking to you. So is there anything else that you'd like to hand over to the group?
There is something that I'd like to bring, because we've recently got to do some really cool work, and I'd love to talk about it for a minute. But I also want to ask, bring it to the group. So when BioIVT purchased Xenotech, we'd been working through a digital transformation journey for technically for 5 years, if you count the time I was asking for money from people, but 2 years of real development and validation. We were switching from paper records because we're regulated. We have to meet certain compliance standards. So we were validating and introducing this electronic lab notebook. We did it a little bit differently than everybody else, well, most people. And I know we did because the company we were working with had never done it this way before, where we wanted an end-to-end solution where we literally do the experiments in the ELN and the data comes out client ready, QC checked and all of that, with beautiful curves and stuff ready for the client. That's what we were trying to do. And it so happened that we were acquired by BioIVT right at the tail end of that, which was perfect for BioIVT leadership because they were really of a growth mindset and we could suddenly introduce this. So what we did from that was we kind of pivoted a little bit and we grew into, we recently launched service suites. That's what we did, in vitro services, in different tiers. So tier 1 through 3 in custom. Our old work was very much high content, sort of regulated. You're going to have all the bells and whistles, and so we couldn't meet client segments who didn't fit that. So with the growth mindset and the leadership support of BioIVT, we were able to make that leap and start working again with some of the other clients that I kind of missed because I like working with small and mid-sized pharma more. You get more scope for using your brain in that environment. And I wondered when we get into Q&A how this team is thinking about market segmentation and how they're thinking about that with their products and the work they do. So I was curious about that.
I think that's an interesting thing. I mean, the way I would sort of think of that is it's how thick the package is that you would build. So sometimes you can't afford a thick package, so you have a thin package. And then but and you'd love a thick package, but you can't afford it. So then you have
a middling one is fine.
Yeah, so it's
and it'll meet the need.
It's which is very interesting, because in agile thinking, it's a minimal viable product, isn't it? It's what is a minimal viable product, and I think that's a very interesting question always to a scientist. I've had many battles with many scientists over the years about being very, very thorough.
Yeah.
The whole essence of agile thinking is prototype, explore, test, really, rather than complete, build, put in everything that you want. So I mean, maybe what we're saying here is that in order to, there is a sort of it's a minimal viable package, isn't it? What's the minimal viable?
What's the value for you that you need? Yeah, and honestly, that is so true. I remember my first boss at Xenotech. Then I was doing MedID. I'm an advanced mass spectroscopist, right? So fresh out of school, fresh out of doctorate. And I love what I do, and I love structural elucidation. And there's one metabolite I just cannot quite get the structure. I've narrowed it down, but I can't quite get it. And I'm messing with this for days and days. And finally, my boss at the time, who is 20 years experienced and much more pragmatic at this point, it's like, we've done all the hours on the project and some more. It's one metabolite in a rat out of 30. It's going to be okay. You've got to stop and move to the next project. And that was really hard for me as a baby scientist.
I think that's a really interesting question about cost base and about whether people can afford things.
And is it worth it?
Yeah, because they over egg the pudding. You know what I mean? Yeah. You do it to death until you've got something. I think I've so enjoyed this and would love to talk to you more, but I think I'm going to open it up to general discussions.
Yeah. Should
Graham,
we just say a thank you first of all? Thank you very much, Joanna.
It was fantastic.
I'm going to wrap up the recording.
Let's give you a round of applause. And what we'll do is Graham, you'll do your thing.
I will.